Principium, Beauty Systems and Innovations. Integral ingredients, a fine-tuning of cell physiology. Very often, in our research for biological activity, we like to either activate or inhibit physiological processes. In the same way, hyaluronic acid and hyaluronans are molecules that get only considered for their active properties or passive properties. Get better and subtler reactions. Research carried out on high-tech full-spectrum hyaluronan demonstrate higher and more refined results by rheostatically adapting cell functions to the signals of their local microenvironment. Skins are different, and so is the native pool of components that is inside of our skin. For example, hyaluronan. The molecular weight can vary according to our physiological state. At a normal physiological step, hyaluronic acid is in form of high molecular weight. It gets reduced to a small molecular weight when attacked or under stress conditions. So why only use one molecular weight? You could use a full spectrum to get a full activity. Integral technology is a new approach to sodium hyaluronate. Instead of focusing only on the molecular weight, the focus is here made on the efficacy of the product. Biology is not chemistry. A same inky name can have two antagonist effects. For example, CD44 receptors present on the majority of our skin cells are receptors that are sensitive to the hyaluronans. If the hyaluronan is with a low molecular weight, then a state of activation takes place. If we talk about a high molecular weight, in this case, we observe a state of inhibition of the physiological processes. Let's see how, through the same receptor CD44, we can lead to two completely different reactions with this mechanism of action. Normal conditions. Step 1. Membranoir receptor CD44 awaits to bind with extracellular matrix hyaluronan. Step 2. High molecular weight hyaluronan binds to CD44. This activates phosphatase PP that hypophosphorylate merlin. Merlin binds now to CD44. Step 3. Merlin displaces ERM proteins from CD44 site and leads to their hypophosphorylation. Merlin's CD44 complex starts inhibiting RAC, that in turn starts inhibiting cyclin D1. As a final result, CD44 Merlin complex separates from cell cytoskeleton and cellular activity turns to antimitotic mode, inhibiting skin cells proliferation. Step 4. Transition to stress condition phase induced by binding with low molecular weight hyaluronan. Stress conditions. Step 5. Low molecular weight hyaluronan binds to CD44. This deactivates the phosphatase PP, which results in the phosphorylation of Merlin by PA key activity. Activated Merlin reactivates in turn dormant ERM proteins through phosphorylation by membranoir GFR. Step 6. Phosphorylated ERM complex binds back to CD44, simultaneously displacing hyperphosphorylated Merlin. ERM CD44 complex then activates Rho family. Step 7. Rho family phosphorylates in turn the actin cytoskeleton that comes to bind with CD44 receptor. Hyperphosphorylated Merlin activates RAC enzymes that stimulate cell proliferation, mitosis, through cyclin D1. Step 8. Actin cytoskeleton and Merlin both stimulate ERK that further activates cyclin D1 together with RAC. As a summary, high and low molecular weight hyaluronans influence cell physiology in an opposite manner through their distinctive connection to cell membrane receptor CD44. High molecular weight hyaluronan maintains cell homeostatic stable state, when low molecular weight hyaluronan stimulates cyclin day 1 activity and CD44 cytoskeleton binding, both leading to enhanced cell proliferation, mitosis. Skin cells will therefore react in different manner if they bind to low molecular weight or high molecular weight hyaluronan. CD44 receptor can feel the physiological state of the skin by acting like a little radar for the local microenvironment of the cells. Instead of being forced to do one action linked to one molecular weight, they are now able to interact with the cells and their local environment to do an ensemble of actions. More than an all-or-nothing mechanism of action, the proportions between high and low molecular weight will determine a rheostatical answer for an adapted answer. Specific spectrum, specific ingredients, specific applications. Skin cells are able to sense the initial stress and react in consequence. We selected the efficacy of every spectrum through a screening test. You can see as an example here that 4 different spectrums give 4 different efficacy. Even when compared to classical sodium hyaluronate, improved performance can be remarked. For example, penetration. Skin penetration is actually higher when compared to low molecular weight or high molecular weight. Integral ingredients even demonstrated specific performances, like antioxidant effect or neosynthesis of elastin. One spectrum, one message, one targeted application. Every integral ingredient is unique and shows a unique profile. For example, difference is an ingredient that has been tested for long-term anti-aging. Anti-wrinkle tests are available at 15, 30, and 60 days. Efficacy to the power 3. Cube is another ingredient that concentrates on 3 effects: skin lifting, epidermis health, but also penetration of other active ingredients. Forget about molecular weight. Focus on efficacy. It's time to pass to the 3rd generation of sodium hyaluronate. You can now control and fine-tune skin cell physiology. Get a broader choice than just inhibition or activation. Today, you can fine-tune the physiology of the skin. It's a matter of principles. Principium. It's a matter of principles.